COVID-19 has changed the way we do businesses, the way we live, the way we work. It has also brought about several points of intersection across various industries, multi-sector stakeholder engagements, all aimed at collaborative innovation. The world now stands at the cusp of a revolution, the fourth industrial revolution, where physical and digital technologies are changing the way we live and work. While previous revolutions were often localized, today's is more interconnected and global, and its impact is already visible in homes and businesses across the world. The new India has already set itself on this new path of innovation, a path driven by creativity, innovation, and favorable policy infrastructure. India is the third largest biotech destination in the Asia Pacific region. The world is now looking at India as one of the emerging hubs of biotechnology. And the new India is ready. It is ready to take on the challenges and come up with agile innovations. It is not content with being the back office of the world anymore. As data and information become more and more important, the data encoded in our DNA and other biological substances, the field of genomics and genome sequencing will play a critical role in the future of humanity. Be it preventing life-threatening diseases, precision medicine, or food security, the scope of genomics is huge. With that, I would like to welcome everybody joining us from different parts of the world to the first webinar of the Biotech Spotlight series on genome sequencing by Invest India, the National Investment Promotion and Facilitation Agency of the Government of India. Without further ado, I would like to invite upon Ms. Srividya, Sector Lead, Biotechnology and Pharmaceuticals at Invest India, to give her opening remarks. Over to you, Srividya.
Thank you so much, Apurv. Once again, thank you all for joining this first edition of the Biotech Spotlight series on genome sequencing. As you all know, I think my colleague has also covered about the biotech sector. I would like to quickly touch upon the work which Invest India is also doing, and from the impetus by the government on this sector. So as he said, that the biotech sector has been identified as the key growth segment of the Indian economy. As we all know that India is expected to emerge as the major contributor to the same. And Invest India has also identified some of these sub-segments within this sector as the key sunrise segments, and is equally committed to supporting its growth. Invest India has institutionalized a highly empowered quality team, which has the relevant industry and the commercial expertise to focus on some of these segments and building on these segments. As we're going to discuss this today, and one of the major segment we are going to discuss, which is the genome sequencing, and where we actually see that there is immense potential. But what we have seen that it needs to be further boosted with private and government and academic participation. We've already seen a lot of work which is being done by the government and the government bodies, including Department of Biotechnology and other autonomous institutions, who have really supported the entire journey in the sector, both on the R&D and the innovations front. And definitely, we have seen the results of the same. I think over the years, we've seen the biotech industry registering a very, huge growth. But I think what we need as an industry is further more trust on the same. With the entry of some of the large Indian and foreign biotech players into India, we've seen that we're really able to take that leap of faith in this sector. But I think to further build on this momentum, Invest India has also given more stress to this particular sector, and not only for attracting investments, which is the core of our job, but also of this, of the importance of this particular sector in terms of the contribution it makes to address some of these human health related problems in a major way. So while I said, there is a whole initiative which the Invest India team has taken, but at the same time, we've seen a large part of it, being, a lot of support being given by the government itself. And the government has been able to provide the right impetus to this sector, especially with one of the recent initiative which has been brought into practice, which is the empowered group of secretary and project development cells, which have been set up across the ministries and departments of Government of India, which is focused at attracting and retaining investments into some of the top priority sectors. So these project development cells, which have been set up in various departments, are focused on expediting investments through a very focused coordination with the central, with the union government, state governments, and all industries. Which is basically, supporting all the core investment and, R&D, supporting investment and R&D activities, and building the kind of pipeline of investable projects in the country where, foreign and Indian investors could look at. I'm very happy to inform you all that there is also a project development cell which has been created with the Department of Biotechnology in collaboration with Invest India, which is supporting it, in faster issue resolution, extensive outreach, and policy advocacy activities. So, without taking much time, I think we all know that with the kind of proven quality, scientific, and manufacturing capabilities we already have in the sectors and other sub segments of this particular sector, we believe that we are at a juncture where we feel that the enhanced collaboration with industry, academia, and industry, and government, we can really achieve the vision of the Atmanirbhar Bharat in the truest sense, as envisioned by our honorable Prime Minister. And definitely sessions like these Biotech Spotlight series, which is focusing on the showcasing the success of this segment, is truly a testament to such collaborations. So really looking forward to hearing your thoughts today. Thank you so much. Back to you, Apurv.
Thank you so much for those wonderful opening remarks, Srividya. Now, I would kindly request Dr. Arindam Maitre, Associate Professor, National Institute of Biomedical Genomics, to share his thoughts on the Indian and global landscape of genome sequencing. Over to you, sir. Sir, you're on mute, I think.
Sorry. So thank you very much. Sorry for the hiccup. Let me start by sharing my slide. So I hope my slides are visible.
Yes, sir. We can see this.
Thank you very much. So thank you for inviting me to give this participate in this very exciting event of Invest India, and the role of we'll be discussing genome sequencing, which I understand is very close to my heart, and I'm sure it's close to the heart of my other fellow participants in today's panel. So I will, without further ado, I will just talk about what I have to say on this topic. So I would talk to you about, so the globally genome sequencing has unfolded over the years since these approaches to sequence small and large genomes were set up. We have seen a spectacular over years. We have seen how each and every genome of an organism has been unraveled. I would actually focus on something like that is we work on, which is like the role of human genomes sequencing efforts, which India has participated in or is currently undertaking, and how, in terms of the global scenario. This I come from an institute called the National Institute of Biomedical Genomics, which is very close to Kolkata, the city of Kolkata, and we have a sprawling 30-acre campus, which I would like to invite every one of you who are joining today's event. When things are better, you can surely come over and visit our center, our campus. Now, when I talk about genome sequencing, I mean, there are a lot of initiatives India has participated in, and I would like to focus on a few of them which have played a major role at the international level. So after the initial human genome human genome project was set up, India at that point of time did not participate for reasons which are quite well known or not so well known. India did not decide to participate in the human genome sequencing project. Subsequently, there were one or two other major genome analysis projects which also kind of went by because multiple reasons. We were trying to understand what are the modalities of participating in such large international endeavors where we have to join and share our own data, and share expertise. Second, the genome sequencing infrastructure was still in its infancy, although there were initially some initiatives taken by one or two nodal institutes, including IGIB and CCMB. IGIB is in New Delhi and CCMB in Hyderabad, who had taken some initial steps to undertake large-scale sequencing, but really at that point of time, we were not there in terms of sizably contributing to such large international genome sequencing endeavors. The first major endeavor of international genome sequencing initiative where India decided to participate right from the word go was the International Cancer Genome Consortium. So, and to understand what happened, why did International Cancer Genome Consortium happen and why did India participate, I need to go back a step and tell you what we know of cancer. We know now that cancer is a disease, essentially a disease of the genome, and each cancer genome is unique, not only for that particular cancer type, but also for individual patients of the same cancer type. We also know that in the genome, these mutations which drive this formation and progress of cancers happen because of errors which creep in right at the beginning of the evolution of the cell, and in multiple somatic events, these errors happen, which are mostly driven by environmental factors. Now, among all these mutations that happen, the vast majority are kind of passengers in a vehicle. They have no role on how the bus travels. So similarly, these mutations have no role on the initiation of progress of cancer. Only a handful of these mutations actually are the ones which initiate and drive cancer. And hence these are called as driver mutations, and the genes they reside on are called the driver genes. So they're only a handful. And in the vast majority of cancer types are sporadic. That means they don't have a hereditary risk. These 90% of these cancer cases, these driver mutations play a role. Hunting for these driver mutations are more like hunting for needles in haystacks. And that is why mankind as a per se never made much headway into identifying these exhaustively, these mutations that drive cancer, because we did not have the wherewithal a decade ago. So over the last 1 decade, we had we got ourselves the wherewithal, developed the methods by which these kind of mutations can be identified in large-scale genome sequencing. And that one reason is when earlier we were actually sequencing, we are peeking at peaks, so as to speak. We're using Sanger sequencing approaches. We are looking at multiple peaks, trying to assess in a very unquantitative and lower sensitive approaches where we couldn't even detect most of these mutations in the tumor. But subsequently, when something called the next generation sequencing happened, instead of looking at peaks, we could count. And that transformed our understanding from qualitative to a quantitative assessment. And we could count how many got sequence reads instead of peaks. And we could count how many reads have the mutation versus how many reads which do not have, and hence could use quantitative measures to identify these mutations. This technological breakthrough resulted in formation of the International Cancer Genome Consortium, whose sole goal was to obtain a comprehensive description of the genomic, transcriptomic, and epigenomic changes in at least 50 different tumor types globally. So I'm talking now of the global picture, which are of clinical and societal importance across the globe. And for each of these tumor type, we wanted to sequence about 500 tumors, which comes down to 25,000 cancer genomes. And for each of these genome, each patient, we would actually sequence the tumor genome and sequence the normal genome from the same paired normal genome from the same patient. For most solid tumors, it was the genome was sequenced from the blood. And so that is how the Cancer Genome Consortium took place, so this is the sole goal of the Cancer Genome Consortium that happened, and that's how it unveiled. So hunting for somatic drivers. So this is the concept that the Cancer Genome Consortium kind of was the holy grail of Cancer Genome Consortium, identifying these somatic drivers by searching for these various genomes. We look at paired tumor and normal DNA, and then we identify the somatic mutations. So today, this Cancer Genome Consortium, which has sequenced 25,000 cancer genomes, say that no cancer therapy is developed today without the genomic knowledge that ICDC provided to the world. And that's a proud claim. And if you look at how the cancer genomics has unfolded and how some of these marvelous discoveries of the whole genome sequencing and cataloging has translated into reality in the clinics globally, you would appreciate that this is not an empty claim. So India, as I said, participated initially as the founding member. India decided to work on gingival oral cancer. So that's the unique subtype of cancer in India, and that's caused primarily by chewing of tobacco. And hence, this initiative was fueled by funding from the Department of Biotechnology, Ministry of Science and Technology, Government of India. The various secretaries during that time they played a very keen role in supporting this initiative, and this was primarily spearheaded by National Institute of Biomedical Genomics and Advanced Center for Treatment Research and Education in Cancer. So together we worked on this cancer genome, and we have sequenced close to about 500 tumor genomes of gingival oral cancer today from India. And these are the various outputs that have come up. I mean, if you look at over these kind of scenes, so this was for the first output that came up from this sequencing of this large cancer genome was identification of drivers of gingival oral cancer. Then we looked at mitochondrial genomic alterations. We looked at specific pathways, and then we looked at risk for lymph node metastasis. Some of these genomic mutations could happen. We looked at epigenomic and transcriptomic alterations and how gene dysregulation plays a role. So, and many of these, and finally this culminated in 2020, a large-scale ICGC TCGA pan-cancer analysis of whole genomes consortium, where India also participated with the oral cancer genomes, and this was published in Nature in 2020, which is kind of very highly cited. And still people are working on the data and what it means and how this can be translated. A fraction of these discoveries can be translated to the clinics. It will completely transform the way cancer patients are managed in a genomic governed fashion. So these we are still working on some of these even after we have identified major things, and we are actually, to give an example, out of an output for this large cancer genome initiative, we have already published a database of oral cancer mutations, exhaustive database, and we are presently working on a sequencing gene panel to start with on these translational tools that can come out, which can predict risk for developing lymph node metastasis or from pre-cancer to oral cancer. So these are some of the examples we had been working on in oral cancer. So this is the Cancer Genome Initiative, and this is how vis-a-vis the global situation, India played a deep role. The second thing that I would like to talk about is population genomes. Now, over the years, understanding genomes of different populations have played a very important role. So in terms of, if you started from the HapMap project that globally the scientific community started working on, went down to 1,000 genomes project where more and more genome sequencing started playing a role. And now each and every country have initiated their large scale population genome projects, which means they're cataloging their population in terms of the genomic variants, identify what are the population specific genomic variants which might be important in health and disease. So in India Government of India has launched this initiative called the Genome India. In the first phase, it is working with 20 institutions across India. It's a challenging ask. And it is in the first phase, it is looking at sequencing these 10,000 genomes. So all the major players like NIBMG, IGIB, and CCMB and other partners are actually playing an important role in generating these data sets. These are to address the global problem that we presently have of Eurocentricity in studies of human genetics. The Indian genome is not adequately represented, and that results in sizable difficulty when we undertake these health and disease genomics studies, large-scale studies in Indian populations. So this is essentially the unfolding right now, and in very short period of time to come in future, you will see marvelous outputs from this large-scale genome initiative that is happening even in the first phase. So the third genome aspect that I can talk to you about is the viral genome sequencing. The viral genome, so as we all know, that globally we are stuck by a pandemic of SARS-CoV-2, and India, the outbreak started early last year. And right from the beginning of this pandemic outbreak, NIBMG and many other centers in India had been actually actively working towards cataloging the SARS-CoV-2 genomes that are present in different individuals who are infected with the virus, to understand how the infection has been introduced, how it is unfolding as the pandemic goes on and rages on between first wave and now we have the second wave, and we are wondering about a third wave. So right last year, NIBMG coordinated with a few other DBT institutes primarily and set up the first 1,000 SARS-CoV-2 genome sequencing consortium, where we actually collaborated with clinical hospitals in multiple regions of the states. On the left side, you can see that there's a various states where we actually networked out, and we generated the catalog of first 1,000 SARS-CoV-2 genomes, and we rapidly made it available in the GISAID for the global community and Indian scientists to actually work on and use that for working further on the virus and how to control the pandemic. Subsequently, Government of India has launched a very structured, organized, large-scale genome sequencing consortium called the INSACOG, the Indian SARS-CoV-2 Genome Sequencing Consortium, which is coordinating initially with 10 sequencing laboratories and National Center for Disease Control. Now it's expanded to 28 laboratories, and then we are also now networking with specific hospitals across the country in sampling not only the sentinel surveillance, where we are sequencing a certain percentage of the daily positives, but we are also focusing on sequencing the genomes of the virus from the nasopharyngeal and oropharyngeal swabs from patients who actually had to be admitted to the hospital. So to sample both from the general population as well as from the hospital. So the objective is to determine the genomic variants and to identify any surge that might be related to a particular variant. Of late, many of you know that the Delta variant that was right now is the primary driver for the pandemic is actually first spotted by INSACOG when the surge cases happened in India. So right now, I mean, globally, the scenario in the GISAID, India has actually sequenced more than the initiative has sequenced more than 60,000 genomes of the virus, spread across multiple regions of the country, and now in a more uniform manner. And already about 42,000 of these genomes have been shared in the global GISAID for the entire community to work on this viral genome information and all. Now, these are some of these initiatives that I would have liked to give example. I will rapidly end with the infrastructure. Understand that genome sequencing or genome analysis requires sizable infrastructure. Initially, we could not participate in many of the major genomic initiatives, primarily because we lacked the infrastructure to actually contribute. But over the recent years, we have now are getting into a position where our genomic infrastructure has developed, and as an example, I can provide you this National Genomics Code that has been initiated by Department of Biotechnology, Government of India, where this National Genomics Code is basically to provide genomic services to the academic centers in the country as well as to the industrial sector, so that anybody has a requirement of a genomic service like genome sequencing, large genome sequencing, say, or small genome sequencing initiatives, or genomic related services, then this National Genomics Code houses the best of the infrastructure, and also is run by professionally trained manpower, so that these teams can actually generate the data, provide the answers to what these academic or industrial users would like to have. So we are right now, the central hub is in an IBMG, where we have multiple NovaSeq 6000s, which are the highest powered workstation for genome sequencing today. We are providing a large amount of executing a large number of projects. We have decent data analysis and storage infrastructure parallelly that needs to go hand in hand in developing a genome centers. And this is how together we are taking on large projects, like contributing in large projects like the Genome India or in the INSACOG or in other large projects. We are actually contributing this manner. So this is what the National Genomics Core is providing in various aspects of genomics related work to the entire country and to the industry. So I will end here. Thank you for giving me the opportunity and listening to me. I'm always happy to hear. My email is here for everybody to use. Anybody wants to know anything further on this, they can drop me a line. I will be happy to answer. Thank you very much.
Thank you very much for that insightful presentation, Dr. Arindam. You touched upon the next generation sequencing. I'd like to request Dr. Rajesh Pandey, Principal Scientist, Institute of Genomics and Integrative Biology, to share his insights on the next generation sequencing. Over to you, Dr. Rajesh.
Thank you, Apurva, and thank you, Invest India. And let me start by saying hello to Arindam also, because I am seeing him after some time, and I have known him for quite some time. So good to see. And also, you made my task much easier, because you touched on all the relevant topics of genomics aspects of India, led by NIBMG, wherein IGIB and CCMB has been partners. As I kind of prefaced initially, that I am not probably going into the nitty-gritty of it, but possibly share my experience. And when I say my, it is through my institute's experience, which is IGIB. So incidentally, IGIB, which we now know as Institute of Genomics and Integrative Biology, was actually christened as Institute of Genomics in the year 2002. And I joined IGIB in 2004. So I missed the initial 2 years, but I have seen the journey of IGIB as a genomics institute, and how it has learned through the process of initial days of genomics, and what it does now is how it has reached that stage. So when I got baptized into genomics, I had 5 Ds which were kind of very important for me, or when I thought that probably genomics would provide answers to this. And those 5 Ds at that point of time, which are probably relevant today as well, are discover. So genomics gives us an opportunity to discover something, which Arindam mentioned very well in terms of how, in terms of cancer, the somatic mutations, that is possible using the strength of genomics. So once we discover, the next I thought would be relevant to develop. Now, what do we develop into? So if we identify a group of SNPs or group of mutations or group of variations which may be having a prognostic or a diagnostic value, then they can be developed into genetic assays. So from discovery, we can make a subset of development of genetic assays. Then we can use these genetic assays to detect something. So we can use initial set of, say, sequencing 1,000 samples of either human or any particular pathogen, and then we can use this assay to detect something in a more cost effective and a more high throughput manner if we can convert it into an assay which can be done faster and also cost effective as well. So we have now covered 3Ds: discover, develop, detect. Now, after this comes the fourth one, which is diagnose. So can we use this and translate this into a clinical setup as part of either a hospital setup or as part of our, path labs, where this can be used for diagnostic purposes, and we can say at least with a broad, a good level of confidence that this particular patient subgroup or a patient subset may have a not so positive outcome. So we need to closely follow over a period of time. And if this all goes well, then probably we also add the 5D, which is the drug development, that at the end of the day, even today with SARS-CoV-2, when we have diversity of symptoms from asymptomatic to mild, moderate, and severe, at the end of the day, when a patient arrives in a hospital, the patient is treated for the symptoms. And that symptoms is alleviated by use of certain combination of drugs. So we thought that these are the 5Ds which are quite important. But in last 15, 16 months, when India was not, left untouched with the SARS-CoV-2 pandemic, we also realized very closely that probably to add a 6D to it. And that is that we need to democratize genomics more aggressively and more proactively than possibly before. So one IGIB, one NIBMG, one CCMB are probably, we need to make at least 5, 10 clones of this at different scales, at different places, at different, for different requirement. So this journey of 5D to 6D is something which has happened quite well. But to achieve this objective of 6D, wherein we are talking about discover, develop, detect, diagnose, drug development, and democratization of genomics, there comes another S, which is very important, which is, I call it as 4S. That is, we need scale. We need scale of sequencing. When I say scale, it means both ways. We need to sequence faster and we need to sequence even high number. So we need scale. We need speed of sequencing as well, that we can't, the sample to sequence gap should be as low as possible so that we increase our chances of discoveries much better so that it can be used proactively for prevention downstream. So the second is speed. At the same time, sequencing or genomics is also very importantly defined by sensitivity. So our data should also be globally competitive. So we can share that data globally, so the data needs to be sensitive. And fourth important aspect is per sample cost as well. And that is especially relevant for a country like us, wherein we have a larger population base. And when a pandemic like this strikes, we need to also actively consider that can we lower down our per sample cost without compromising on the efficiency and the sensitivity of the sequencing output. So then comes the 4S of scale, speed, sensitivity, and sample cost. So as we move through this last 15 months, and I keep on citing this last 15 months because having seen closely, this 15 months has taught all genomic scientists probably the learnings of last 10 years, wherein we learned how to respond and how to work 24/7 and how to respect the mandate given to us in terms of the expectation that we would be providing leads which would be followed up as we move on. So we needed a combination, which was like we needed both high throughput sequencing and also high priority sequencing. So we call this concept of high throughput sequencing as driven majorly by MegaLab, which is powered by high throughput sequencers, which are in the mentioned, which is powered by NovaSeq 6000, even iSeq 2500. But at the same time, we also require high priority sequencing. So if there are specific clinical subsets which is behaving, or as per clinician, if it is behaving differently from the other in the same area, we need to sequence it quickly as well. So we need high priority sequencing, and we call that as a micro lab. So micro lab means a lab which can be set up with ease in resource limited settings with probably lesser requirement of a hardcore infrastructure. So the idea of this mega lab and micro lab, I'm not going deeper into it because we have publications online which can be looked into, that how do we define micro lab. But basically, by when we say micro lab, it means a sequencing facility which can be developed or implemented in a cost effective, low resource settings. And one example of which IGIB did quite successfully when alpha variant was staring at us was we set up a mobile sequencing at the Delhi airport. And that's basically my director called me at 10 in the night and we set up a sequencing at 10 in the morning next day, wherein we just packed the things in a car and went to the airport and set up the sequencing in a mobile van. Mobile van. So that's the strength of building up the micro lab network. So how did these things happen? And good part is, or the positive part of the story is that there has been good progress in this direction, and there is a continued progress in this direction. So how it has we have achieved what we have achieved. So what we saw very closely was that there has been a thinning of boundaries. And when I say thinning of boundaries, it has been thinning of boundaries on multiple aspects, which is primarily even between academicians or scientists, there has been absolutely free flow of information, free flow of ideas, free flow of notes, free flow of experience to see that how fast and how better we can do what we want to do. Then there has been seamless connection between the clinic, because whatever we want to do as a genomic scientist through next generation sequencing, clinicians need to be at the core of it, because they are the ones who are going to provide us that yes, this patient subgroup is something different, although they are all SARS-CoV-2 positive, for example, but their symptoms are different from the others. So there is a case for us to know that is there anything in the pathogen or in the human host per se which is making it behave differently than the normal. So there has been an active participation or coordination between clinicians pan India. And the third and the important part is that whatever we do, we need some funding to do it. And the funding has been quite well in terms of both national, international, canonical, non-canonical funding. So that has been also, led to a thinning of boundaries on these three broad parameters between academicians, clinicians, and the funding bodies. Now, that is a good part of the story, or the positive part of the story. But then this needs to be sustained, because we can't allow ourselves a situation where we again start our learning from, say, going to step back. So one of the way which we see that it can possibly sustain itself is we keep on building a hub and spoke model as much as possible. That doesn't mean that there will be one hub in India and then all would be spoke. So we need to have as many, as Arindam mentioned about genomics course. We can have as many hubs catering to a small catchment area, and then we can have spokes. So how do we visualize that it can be a good idea to build on it, that possibly say the hubs would be a combination of mega lab and micro lab, wherein hub would have both the strengths to do, and then the spokes would be essentially a lot of micro labs. So we may be thinking that why this model would be important, or how this model will work. So the institutes which have, say, made their learning in last 10, 15, 20 years in terms of genomics, we also have a role in terms of being now in a position of enablers. So we need to handhold the new partners into genomics and say that, hey, we have a SOP which you can practice from day one in your setup. We provide you the infrastructure for analysis till you build up your own infrastructure, and we conduct training programs, because that is integral for the success of it. Because once we have a trained manpower, both experimental and analysis, they will be the workforce who will implement or guarantee success on the ground. So it would be a hub and spoke model, which would be based on benefits for present for sure, with deliverables earmarked, as well as highlighting to prospective stakeholders as well, that well, this is a model, or this is a system which is working perfectly well with the outputs which are of relevance for present and implications for future. So that's one aspect we think. And the next one, which I think, which was also progressed quite positively, is that there has been a lot of opening of boundaries as well. So one of the positives of, I mean, with a lot of other pushbacks or negatives from the SARS-CoV-2, but one of the things which has been positive that is that we had a lot of proactive interactions between the stakeholders globally, nationally, who want to make their, pool their effort, pool their resources towards having a more quality inference in a quicker time period of time. So there has been a lot of opening of boundaries through a lot of virtual meetings, through WHO, to whatever we can name. They are all partnering with each other to see that how the insights can be shared and the inferences can be transferred to each other in a quick time. So all these learnings, how do we see the picture beyond what we are at point of time? So there is something which is an idea which I would park here as a part of Invest India, and I will look forward to having more discussions on this regard as we move along. Is something if we can develop something called I say it as GPS, which is genomics-based pathogen surveillance. So SARS-CoV-2 is not going to be the pathogen, the only pathogen which has caused what it has, and it is not going to be that way as well. And we see around us that there are a lot of pathogens still. They haven't kind of vacated the space for SARS-CoV-2. They are still there. It's just that we are kind of, the other one is has swam us more with more strength, so we are responding to that as well. So can we develop localized plus national level genomics-based pathogen surveillance, wherein again one person, one institute probably are not good enough to have an impact. So we need partnerships. We need partnership across expertise, which includes researchers, which needs genomics people, which needs clinicians, which needs investment partners, which needs public funding, private funding, grants, foundations, non-canonical, whomsoever are welcome to join hand, and I think that's why this forum is quite important, wherein we need to have that conversation and communication to see how can we sustain the manpower which has been trained in last 15 months. So we have a much bigger pool of genomics person now, wherein we have people who are more better than in the last 6 months even, who can do more sequencing, who can interpret the genomes, who can do the analysis thereafter, who can liaison with the clinicians to also highlight to them that why it is important for a clinician to also partner with a genomic scientist and how the findings can be translated back to the clinician. So we need that communication, sustained communication as long as possible. My wish list is to continue forever. But again, I leave it. I would stop here, but I take this opportunity to have initiate a conversation that can we have programs, prioritize few diseases which are there. MTB always there since my start of scientific days. I always hear about MTB, but can we have a genomics-based pathogen surveillance program for MTB? I know some offshoots have started, but can we have as pervasive program as we are now doing for SARS-CoV-2? And it is almost a record that in last year and a half, we have 2.5 million SARS-CoV-2 genomes. And probably half of this is lying in different labs who are in the process of uploading to GIS8 as well. So if something like this, we can definitely do a quarter of this for other pathogens who have a much bigger impact on overall, the health infrastructure and the health quality of life. So with this, I will stop here and thank you, everyone. And I think we need to have more discussions around this and see how we can define problems, make progress towards solving those problems with deliverables. So probably the problems cannot be solved open-ended. We need to define goals. We need to define deliverables, and then going through a solution-oriented approach. And with that, I will stop. Thank you.
Thank you so much, Dr. Rajesh, for your very intriguing comments. It was really interesting to learn about the 6Ds and the 5S in democratization of genomics. In your session, you mentioned the need for collaboration with private industry and investors. And for that, we have Mr. Sumit Jamwar, Chairman and CEO of Global Gene Corp with us today. Would kindly request him to share his, how his company's experience has been in India, and what they foresee for the future of genomics in India, and how their role would be for key to this collaboration with the country. Over to you, Mr. Sumit.
Thank you, Apurv. Look, first of all, thank you to Invest India for this invitation. You have been a sponsor for our work within India, and you have been an amazing partner. So we really appreciate all that we've been able to achieve and the support you've been able to provide. Look, it's amazing to be following Professor Maitra and Dr. Pandey, because both of them we know by reputation. I mean, the work has been phenomenal, and I think I would say that both of them have made my task easy, because I can now just pick and choose certain aspects which need to be highlighted. And Dr. Pandey, one thing, and to both of you actually, one thing I would say up front is absolutely, I mean, both on talent, send us your best talent. We need the best talent, and we will train them, and we will collaborate with you in terms of moving things forward. So please use us as that. In fact, I think one of Professor Maitra's research colleagues actually works with us. So I think, and again, we want to build that, because that's how collaborations and innovation ecosystems work. And the second thing is, again, the pathogen surveillance and in general the genomics world that you talk about, that's very close to our heart. So we are all, as they say, the saying is, we are all singing from the same hymn sheet in terms of what we want to achieve and how we want to achieve it, because we're all very, very, very much scientifically minded. Now, at this point, look, I wanted to take you back to, I appreciate what you're doing, because I was very fortunate to grow up in the academic environment. My father is a professor. He was a professor at the Institute of Technology, Delhi. And so I grew up on the IIT Delhi campus. And you can imagine, as a young kid growing up surrounded by these amazingly smart professors, who all they used to ask was problem statements and questions. That's all they were interested in, right? And which is a phenomenal training, and for all the kids. To actually focus on what is the problem and how you think about a problem, and that scientific curiosity which remains. And I think that's what brought me into the world of genomics. I'm a chemical engineer from, again, I was fortunate to go to IIT. And what brought me back is the fact that with genomics and with the DNA, we have a real opportunity to shape and understand who we are, and actually use that to personalize health and personalize our life experience. The whole if you were to think about it, imagining the fact that we can, based on our DNA, we can live our life to a fullest potential, avoid diseases where possible, and if something happens, then we can personalize the cure against it, or find cures, or in the future use CRISPR and other technologies to actually eradicate those. I think is a mind-boggling possibility, and this is what I think, as a science geek, this is what I grew up to. Now, what I want to do is, with your permission, just share, a few, few slides. I'm just trying to see how I can do this in terms of the window. It's not showing up. Just give me one sec. Just trying to. Just conscious of the time, excuse me. I just wanted to touch on a few points today, right? So the few points that are there, in terms of this one, I think. Right. So what? Okay, are you able to see it?
Yeah. It's visible.
It's visible. Great. Okay. It's the key thing is, look, what excites us is that we are fundamentally excited by genomic revolution. And if there are three messages that I want you to take away, is that genomics is fundamentally changing the health paradigm. But we as Indians, and I would say Asians, are missing out. The second message I wanted you to take away is something that Professor Maitra said earlier, is India is one of the most diverse countries in the world, and the scientific insights can help us create cures for diseases across the world. And more importantly, it can enable the 1.3 billion people to leapfrog to the healthcare of the future. And I'll talk a bit more about what, I highlighted what it meant. And the third element, which is something which my colleagues have highlighted before, is this task is hard. The science and the ethics are fundamental to success, and we will only succeed in collaboration with like-minded people who have a passion. And that's why, again, I highlight the sponsorship from Invest India in terms of the vision and the support that they've provided. Now, again, this is something you've heard. This is something you may have seen. The genomic revolution is transforming healthcare. Look, the cost per genome, what costed $2.7 billion in 13 years, takes less than $1,000 in a few hours. And we are actually, my research team, some of my research team is actually based at the Wellcome Genome campus, which was one of the sites of the Human Genome Project. In fundamental, and this is something, again, Professor Maitra touched on, and again, Dr. Pandey mentioned as well, is genomics plus data is what is transformation. But we think about the Asian population, they're missing from the global data sets. There's no data for, negligible data, you might say, for Asians, who are almost 60% of the world's population. India right now is contributing less than 2% of genomic data and insights. And our mission, and this is why we set it up, is to bridge this genomic divide, right? Building the most comprehensive and deepest genomic data for Asia, which looks at about 6,000 odd global ancestral lines. And the reason that is important is because to arrive at the benefit, we need to have an understanding of who we are. Now, India is one of the most genetically diverse countries, right? Yet it, as I said, contributes less than 2% of genomic data. We are a nation of 1.3 billion people, 4,500 ethnicities. So if you think of the world has about 10,000 ethnic groups. And, this is something, again, was mentioned previously, but 4,500, almost 50% of that is in India. And what that means is that we have a remarkable scientific opportunity of understanding what the special characteristics are. And from that, we can find solutions to, let's say, diseases, or we can also use that information to enable each one of the residents and people of this ethnicity to have a great life experience. Now, the challenge, of course, is that this path, this is not an easy task, right? So it starts at a fundamental level. The first and the most important thing is one thing which was there in a slide earlier, is the ethical consents. The second and really critical aspect is, the science behind it, on defining what the science is. How do we understand and create an understanding of the population? And that's where we are very fortunate to have launched a sequencing program for the populations of Indian subcontinent. Again, we are fortunate with support from Invest India, and this was I've just picked it up from the Invest India website, where we are looking at sequencing the populations to understand the population structure as well as diseases. Now, the reason why this is not an easy task is because it starts at making sure everything is in the right ethical framework. Science is hard, so you have to define what the scientific problem we are looking to solve is. And then execution is hard, because in many cases we do not have structured electronic health records, right? If you don't have EHRs which are standardized, normalized, then we have to build it. And so we had to start from scratch, where we built a tablet-based real-time digitization. So when we recruit participants, we actually do a very detailed phenotype assessment and add the electron, almost creating an EHR, which the bioinformaticians can later on work with that structured data. And that's just one of the problems. And again, we are very happy to extend that capability, because we've spent that effort, and we know it works, and it works in a diverse and large country like India. We're very happy to extend that to colleagues, because it shortcuts the time that people have to do in a variety of ways. Again, these are some hard problems we had to solve to get to where we are. The other element is very much around sharing and around collaboration. And again, I'm emphasizing the point that my colleagues made before. It's about how do we share it. So, we are very fortunate to launch the first India beacon, genomics beacon, called GGC India, for democratizing access to researchers. So where we can, we share it. It was launched by the Deputy High Commissioner of India in 2017. Again, this is linked into Wellcome Genome Campus, Sanger, Global Alliance for Genomics and Health, etc., who are all supportive. So again, when you see those logos, it's all about collaboration and what we can build from there. And again, it's an opportunity for people who want to share and want to engage with it to use the beacon to create that, that sustainable flow around India, so that we can help researchers and also benefit from that on the other side. And then is the other aspect of saying, how do I stay in touch? What is it that I'm tangibly giving to someone as a participant? So in order to keep in touch, not only on the tech stack on how we input the data, but then the analytical platform, and then, engaging with the research community, is the return of results to participants. So GGC Me was created as an app which is in implementation right now, for example, in Singapore, in the Institute of Precision Medicine, where GGC Me, which is provided by us, gives the return of results to the participant. Now, again, this is an opportunity when we are sequencing these people. We're very happy to extend this capability because we know it works. We are giving something relevant to the people who have had access to, who have participated in our studies. And it is a way of saying thank you, but it's also a way of staying in touch with them. And again, this is where the experiences that I wanted to highlight was all around the thing we have found is that technology is a fundamental driver in anything that we do. But that technology has to be localized in the setting that we operate in, like India has its unique characteristics. And in these special characteristics, we need to make sure that we accommodate for those local customs, norms, etc., and use that technology in a way that blends in. And the third aspect, the learning, is the global collaboration. The fact that we have teams sitting in Welcome Genome campus or Boston or Singapore or India is because we know the kind of talent that exists everywhere. We know that we need to train the talent, and we find that fostering of connections is really important. And the same thing we find with other industry partners or other academic partners, where we are in some ways at this frontier science, learning and building that together. So on that note, as I finish, we are very fortunate to be recognized again, thanks to Invest India and thanks to folks, where, I'll just pause here in terms of the recognition and the support and outlining of the vision that has come into play. I would just say, look, we're with gratitude. Thank you for the support. But also it's a long journey. It's a hard journey. And the more collaborators we have working with us and who are our partners, the more fun we are going to have in solving some really important and interesting scientific problems, which will ultimately have impact on 1.3 billion people, extending to about 2.5 billion people across Asia. I just stop there. And on at that point, very happy to take any questions. But thank you again for the opportunity to share our thoughts.
Thank you so much, Mr. Sumit. It's really exciting to see your commitment to India and Asia at large as well. And rest be assured, Invest India and India at large would always be supportive to your operations in India. And we look forward, to this journey that Global Gene Corp has in India, and we hope that the collaboration lasts for a very, very long time, and we're able to support you in all ways that are possible.
Well, thank you, Purn. One metric I would say of success is, hopefully with Dr. Pandey as well as Professor Maitra, we will have some more interesting conversations about what problems we are both looking to solve right now. And please send your best students to us. Thank you.
Thank you very much. We have some questions for the panelists. Dr. Maitra, we have one question for you. So, the question is that with India gaining momentum in the field of genomics, genome sequencing, how do you see India competing and contributing at a global scale in the next 5 to 10 years? Requesting for your brief thoughts on the question.
Thank you. I think it's a very, very strategic question. So as I tried to convey a sense that over the last few years, there has been phenomenal changes in the way infrastructure has been developed in India for genomics, because essentially you must understand that in an area like genomics, it's very infrastructure driven. You cannot contribute unless you have the firepower to contribute. So I think what primarily so far the government and then now the industry is also coming up has decided and realized this opportunity and have been investing. And over the last few years, I think now India is in a very strategically and very nicely poised in terms of genomics expansion, in the sense that there's substantial infrastructure that has gone in. It's continuing to go in to develop the abilities for Indian setups to take on large-scale genomics initiatives. and the fact that we are also developing a large number of, as it is mentioned, that we are using a large number of as Dr. Jamwar mentioned, that we are generating a large number of trained people, because without people, no technology works. So trained people who can do the experimentation, who can analyze the data, who can make sense out of the large scale genomic big data that we are generating in genomics. So combination of these factors, I think India is poised to take over this expansion very nicely in a global scale. Requires investments, of course. Genomics again is a very investment driven sector, but I think now, if the investments happen as they have started happening, I think India is poised very nicely to take on this expansion globally, compete with the global scenario in terms of doing these large scale initiatives.
Thank you so much, Dr. Arindam. As we're short of time, so we'll just have one last question, common to Dr. Rajesh and Mr. Sumit. Both of you touched upon how the industry and academia can collaborate for sharing skills. So, on a larger scale, how do you see how can India develop that specialized skill set as far as genome sequencing and genomics at large is concerned, India being an emerging market? Would kindly request Dr. Rajesh to go first and then Mr. Sumit.
I think it is not that complex, but I think the start needs to be very basic. And again, I go back to very probably the clue for that is in the 80 GCs, which we understand in terms of the genome sequence. So the basics clear that we cannot, start with a, for example, almost all academic institutions, we receive n number of applications for dissertation projects. Now, these dissertation projects can be used as one of an avenue wherein and they normally is for 6 months. So we can, genomics institutes like us, you can use this vast pool of students who join us for 6 months to train them. And I think when I say train, I mean more training, because I still see a bit of a juxtaposition in terms of words of sequencing and genomics, which are not the same. One is an enabler to understand the science of sequencing, and another is a science in itself, which is the genomics. So if we have students at that level who are, say, bachelor's, BTech students, MTech students, master students who join us, we take them not through a take through them a mental learning process wherein we train future genomics people who can understand and appreciate the strength of genomics. At the same time, it is also important to inculcate a seed in the mind that genomics can solve many things, but it cannot solve everything. So I won't be selling a dream that we can solve all the problems in the world by sequencing enabled genomics understanding. But yes, it does provide insights or enables a lot of downstream understanding. And prime example of this is when we are talking about SARS-CoV-2 and all the variants of concern. So genomics identifies the background mutations which are driving these variants of concern, but at the same time, we require complementary skill set to understand that how much of a functional impact those mutations have in terms of their neutralization ability and all. So it's a training of mind which is important. Rest of the things are if I understand or learn the art of asking those questions, I would be better placed either in IGIB or NIBMG or in GenCorp. So it's a matter of how do I train the mental aspect of it. That is more important. Technologies do change over a period of time, and that is what my message to students as well, that do not ask questions from the technology perspective. Today, when I started my genomics experience, it was Genome Analyzer 2. It moved on to HiSeq 2000, HiSeq 2500, NovaSeq, MySeq, then all the other options of AntTorrent and Nanopore and Solid and PacBio. So technology is not what at best technologies are enablers, but if we can learn the art of asking those genomics based questions, define those questions, then we are better prepared for next few decades that yes, if technology changes, my science doesn't stop. I keep on asking those questions where technologies are enabling us to answer those questions.
Thank you so much, Dr. Pandey. Mr. Sumit, if you can
Yeah,
add to
that's a great response. So look, the way, I mean, in a very simple way, the way we look at the genomic revolution is it's a bit like you remember in the 80s and 90s, to get a landline was a 6 to 8 year wait, right? And that too, if you accelerated it, someone knew someone and that accelerated by a couple of years or something. And then the mobile phone revolution happened, right? And right now, if I want a mobile phone or SIM card, it takes me within the same day, I have the access to the best this thing with internet and, phones, whatever you want to do. So now think about the genomic revolution in a similar sort of way. I almost draw a parallel to that from a behavioral change and business model perspective, but I will draw a parallel to internet revolution. Now, when internet happened, the beauty of what we have in India is that we have a highly trained set of people who have a certain skill and a way of looking at things, whether it's in the scientific sort of curiosity, who are graduating every year. And if you take a subset of that in a specialized course like an IGIB or the National Institute of Biomedical Research, when you put those things together, we have a funnel of people that you can create. And it's the same way as we got computer scientists and people who are not even computer scientists who went into IT and other things which started happening. Now, the opportunity I want to leave with Invest India is now, if you were to draw that parallel, India has been very successful in information technology, billions of dollars of return, which has come into play and return and that's generated, soft power for India. That's exactly where we are in genetics. We have the government institutions, so public institutions, we have private institutions. When you look at the combination and we look at the training that we have in terms of trained manpower coming through, which we can either apprenticeship based model or through an academic model, we can train. We are creating an infrastructure which over the next 2 decades and beyond we are uniquely positioned to be the leading country, because we'll give new scientific insights, we will give new business models, we'll give new applications in the way that people haven't seen before. And that is what is truly exciting. So we have all the core ingredients. What we need is the intent and the engagement. I think, one thing is that, being on both sides, on the public side previously when I was growing up and on the private side, I think there's a bridge to be crossed between the collaboration and us building off each other and moving those things forward, because that's how we can utilize the best of our abilities, because the intent is there. It's about making it happen. So think of that mobile phone and think about the IT revolution and how India is playing a dominant role in that. And that's what we have an opportunity to create here today.
Thank you so much, Mr. Sumit. It's been really energizing to hear all of your thoughts, and we really thank all of you, all the respected panelists, for joining us today. I would kindly request Ms. Srividya for any closing comments that she might have.
No, I think we had a great session, and thank you once again to all our speakers, Sumit, Rajesh and Arindamji, for joining us today and making this first edition of Global, the Genome Sequencing Biotech Spotlight series ahead. Thank you so much, and thank you all the audience for joining today. Have a good evening.
Thank you so much. Thank you. Bye.